In this video we will discuss about this bacterial mechanisms, as we have seen that in some of the bacteria got upregulated when they are either treated with this cis-UCA or they are exposed to this UVB. So from the literatures, the researchers find out that the bacteria utilizes or metabolize this cis-UCA using HERC genes. So they wanted to check whether the bacteria that are upregulated will have this kind of metabolisms that they utilizes the cis-UCA using urocanase, which is found by HERC and HERT genes. So first, what they did is the experimental plan is that they grow this bacteria in vitro in different concentration of this cis-UCA and then check the growth rate of those bacteria. And they use two opposite kind of bacteria, that is this Staphylococcus dermatis, and this bacteria has both HERC gene positive and HERT genes positive, whereas Corynebacterium signalare it does not have any of the HERC genes or HERT genes. So for this Streptococcus epidermis bacteria, we can see that with different concentrations the growth rate increases, and for this Corynebacterium the growth rate also increases, but this growth rate comes to a plateau. We can see that from this panel D and panel A. Thereafter, they check this area under the curve. In this area under the curve, they can see that compared to the control, there is a significant increase when they use the cis-UCA at different concentrations. But when they check the area under the curve for this Corynebacterium signalare, here we see that the area under the curve almost remains same for all the conditions, that is for the control and for different concentration of cis-UCA. So indicating that the cis-UCA might help the growth in the Streptococcus bacteria through HERC and HERT genes, or more precisely through this urocanase, whereas since these genes are not present in this second negative control bacteria, we do not see a significant change in the area under the curve. Thereafter, they also check the colonies on the agar plate, and here they see that the colonies in some cases are statistically significant compared to the controls, whereas for the case of this negative bacteria, it is Corynebacterium signalare, does not have any significant changes in the number of colonies. Thereafter, what they did is they want to confirm. They further wanted to confirm. So in that case, they use the inhibitor for this urocanase, that is glycine. And for the control case and the glycine case, they do not see any changes. But when they use cis-UCA and cis-UCA plus this urocanase inhibitor, they can clearly see a change in the area under the curve. And same things they observed for different concentrations of cis-UCA, except for the highest concentration, that is 250 microgram, where they do not see any significant changes. But when they check it for this negative control bacteria, that is glycyl glycine inhibitions in Corynebacterium signalare, they do not see any significant changes in the area under the curve when they use the inhibitors, urocanase inhibitors and cis-UCA at different concentrations. So both these results indicates that the bacteria that are upregulated might have this HERC and HERT genes, and that results in the formations of this enzyme urocanase that can metabolize cis-UCA and helps in the growth of those bacteria, whereas bacteria that lacks HERC and HERT genes not able to form this urocanase enzyme, thereby cannot utilize this cis-UCA for its growth. So this from the two different results, their hypothesis seems to be true, that it's the cis-UCA metabolization using urocanase might be causing their upregulations, and thereby their proportion is high when they are either treated with cis-UCA. So I hope you understood the reason behind some bacteria gets high in proportions when they are treated with cis-UCA, because they utilize this urocanase to metabolize cis-UCA that helps in their growth, and these urocanase are formed in this HUTC and HUTL genes, which are present in some bacteria and which are absent in some bacteria. For example, in this Staphylococcus epidermis has this HUTC and HUTU genes, whereas Corynebacterium signalure does not have this HUTC and HUTCU genes. If you need to, so that's all for this video. If you need to understand more about their experimental results in detail and wants to have a look to their supplemental figures, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.